Welcome everybody to Harrisburg University's Biotechnology Virtual Information Session. My name is Laurie Barrow. I am the Director of Undergraduate Admissions at HU. We're very excited to have you here today. If this is your first time with us in one of our webinars, I welcome you. If this is your first, second, third, or maybe even fourth time joining us, then welcome back. We'll be doing a lot of different presentations this week and over the next couple of weeks and perhaps months. So we invite you to join us time and time again. Today we're going to be talking about our biotechnology bachelor's degree program. And with us today, our very special guest is Dr. Douglas Taylor. Professor Taylor is the program lead for biotechnology, and he's going to be able to share some wonderful information with you today. And once we're finished, we'll have plenty of time for questions and answers. You will notice there is a question and answer section there. It's a little icon with a question mark or like a little box if it's might be already open. We please ask you to ask away questions today. During the Q&A, I'll be able to read through some of those and publish them, and we'll have Professor Taylor answer them to the best of his ability. Just a few things real quick. Everyone is muted on the webinar. You will not be able to actually speak to Dr. Taylor in this environment, but you can ask questions. Also, we have plenty of other webinars that are dealing with things such as financial aid and student services and housing and student support. We're not going to be going into those types of details today. Today is going to be 100% about biotechnology and biotechnology only. However, if you have any other questions about any of the other stuff, you have two resources. Well, three. You can contact your admissions counselor if you know who that is, if you're a high school senior or a college student currently. You can also email undergraduateadmissions@harrisburgu.edu. I'm also going to post a link where we have pre-recorded webinars that you can look at if you'd like to go back and check some of those out. With that being said, we're looking forward to having everybody here today. We look forward to your questions. You don't need to wait until the end to ask them, but we will wait to the end to actually read them. And with no further delay, I'm going to turn it over to you, Doug.
Hey, well, thank you, Laurie. Hope everyone is having a good, safe time off, and I want to welcome you to my basement. It's not quite as cool as Roger Goodell's, but you know, we'll make do with what we have. So today I want to talk about our biotech program, give you some information for some of you who have attended some of our actual open houses. You may have heard some of this, but like I said, this is our first virtual open house. So with that in mind, whoops, can we go to, there we go. Oh. Hopefully everyone can see that now. Is that a yes? Okay, so when we talk about the biotech program, you know, this is a really strange time that we're in now, and one of the things that we really need to think about is, what am I going to do for the future? How is this program going to be presented to me, and what can I do with this in terms of the future? And one of the things, because I'm a parent, I went through this with my kids, one of the things we want to say, if I'm going to be sending my kids to 4 years of college, going through the expense and everything, are they going to be able to get a job and related to the field in which they're studying? It kind of seems like an obvious objective to have, but surprisingly less than 50% of recent college graduates are able to find a job in their chosen profession or in their field of major. So you want to be employed. You want to be employed in a field related to your degree. And you want a job that pays well. And so one of the things I'll tell you right up front, biotech is one of those. Basically, we have 94% of our students graduate and they're able to get a job in the biotech industry. In part because New York, New Jersey, and Pennsylvania are essentially the Silicon Valley of biotechnology. In Pennsylvania alone, the biotech industry employs over 77,000 people, and these are primarily in the drug industry, pharmaceuticals, research, testing, medical labs. Those are the core strengths of Pennsylvania's biotech industry. And on average, the biotech salaries in the state of Pennsylvania are about $103,000. Now, you may say, well, I'd like maybe to explore options outside Pennsylvania. Well, nationwide, 1.66 million people work in the biotech industry at over 77,000 biotech companies. So you may say, well, gee, that's really exciting. Well, what is biotechnology? For those of you who don't know, it comes from the Greek for bios, for life, technos for tools, logos for the study of. So it's the study of living tools. That's really cool sounding, but what is it? What is it really? And, for those of you who haven't heard this presentation before, let's think about some of the famous biotechnologists that you know, and how is it that, and what is it they do, and how does that fit into your career objectives? Well, let's think about Howard Stark. I wore my Stark Industries t-shirt today. Howard Stark, basically, he created the super soldier serum, basically modifying the metabolism of Captain America to become a superhero. Or David Banner, who wanted to tap into the inner strength. How do we turn on that strength that's turned on by adrenaline? So that was one of the ways that he became the Incredible Hulk. Or Will Rodman, basically, he was trying, his father had Alzheimer's disease. How do you cure Alzheimer's disease? That was his goal, looking at neurobiotechnology. That's a course we teach here actually at Harrisburg University. And sort of close to where we are right now in terms of talking about pandemics, Robert Neville in the movie I Am Legend, basically he was a biotechnologist who was trying to develop a vaccine against a bioterrorism virus. And unfortunately, by the time he developed it, the virus had already been released. It had killed most of the world's population or turned them into zombies, and he alone had received the vaccine. Or my all-time favorite, Henry Wu. He basically created all of the dinosaurs associated with Jurassic Park and Jurassic World. So it sort of gives you an idea of the breadth of all the things that you can do with biotechnology. Now, where is it used? How is it used? Everyone, normally when you think biotechnology, the first thing that comes to mind is pharmaceuticals. And certainly 66% of all biotech jobs is in the pharmaceutical industry. But also, we're hearing a lot about that today, diagnostics, whether it's the antibody test, whether it's PCR tests, sequencing, all the things we're doing for the coronavirus, all biotech. Development of vaccines, different types of vaccines, subunit vaccines, synthetic vaccines made in plants, all of these are come from biotech. We all hear about genetically modified foods. That's part of biotechnology does in trying to make crop production more nutritious, better crop yields, and ethanol. In addition to drinking, there's also now for hand sanitizers. Basically, all production of ethanol is the product of biotechnology. One of the things I was kind of surprised to learn until I moved to this area was what a huge input biotechnology has into the manufacturing of chocolate. I know I love chocolate, and surprisingly, there are a lot of biotechnologists that are hired by Hershey Foods. Now, forensics, biosecurity, biodefense, production of biofuels, environmental protection, human health monitoring, these are all under the purview of biotechnology. Now, the biotechnology program, we have essentially 5 different majors. We have general biotechnology, which covers a little of everything. It is very technically rigorous. It's where you learn most, or the application of most of the biotechnology techniques. We have the pharmaceutical drug design, where their students learn computer-aided drug design, pharmacology, bioinformatics, targeted drug design. All of those things are part of the pharmaceutical drug design. We also offer a medical biotechnology program. Includes things like learning medical technology, biomolecules, neurobiotechnology, so how you apply biotechnology to the study of brain and brain disorders, cell biology and its applications, immunology. Now, that said, just as an FYI, most of the people who come in will do better with the general biotechnology degree or focus if they're interested in going into the healthcare field, because it is a more technically rigorous and it's really more appropriate for medicine or healthcare in the 21st century, where we're now talking about molecular medicine, molecular diagnostics, molecular therapeutics. So it gives you a much stronger basis in if you're looking to pursue that field. We also have food science and food safety and quality, basically food and nutrition. We look at how genetically modified foods are created, applied food microbiology, food quality assurance. So if that's something that interests you. Then we have two slightly different programs in nanobiotechnology. We have our in-house program that's a nanobiotechnology where we focus on really the applications, things like biosensor technology, applied nanobiotechnology. We also offer a program in collaboration with Penn State on more focused on nanofabrication, making these materials, nanomaterials, the safety of nanoparticles, and the characterization of nanoparticles. So over the next few minutes of this presentation, I want to talk about some of the hot topics just to give you a feel and get you sort of excited about why you might want to pursue first a degree in biotechnology and then ultimately a career in biotechnology. We all are familiar with DNA fingerprinting. It's the fundamental building block of all forensics is how you identify DNA. Molecular therapeutics, how do you create targeted medicines? How do you create vaccines? These are all based on recombinant DNA technology. Again, these are things that you will learn here. Tissue engineering, that can be creating 3D printed tissues, creating bioscaffolds, creating transplantable tissue grafts. Those are part of the techniques one learns as part of our program. Creation of diagnostics, molecular diagnostics, they can be an ELISA test, it can be the antibody test that we're hearing a lot about, or PCR-based tests. Any of those and how those are created and how they're used and what they can tell you are things as part of the biotech program. Gene editing. That is the backbone of biotechnology. If you come to biotechnology, you cannot graduate until you've edited a gene. That's really an important facet. It is the future of whether you're talking about biotechnology or medicine or targeted pharmaceuticals, is gene editing and gene therapy. And then lastly, sort of my personal favorite, cloning. That's my cat. There's only one of them, but you can clone in this as well, and we'll teach you how that techniques are done. Now let's start with gene editing, because like I said, that's the one that's really critical, and it's a key feature that sort of sets us apart as being unique as a program at HU. Now, you've heard that sometimes you'll hear this term knockout or knock in. That means if you're talking about knockout, that's created when if you have an active gene and I replace it with a gene or piece of DNA that has no function. So basically that functional gene no longer is effective. So you can study what is the effect of that specific gene. Or knock in, this is what we more commonly think about in terms of therapeutics, is a knockout is created when you have an inactive gene is replaced with the correct genetic information. There's currently an ongoing trial where patients who have sickle cell anemia, it's the result of a single nucleotide mutation. And you can take, and what the clinical trial is, they're using CRISPR technology to incorporate the correct hemoglobin gene into these patients, thereby curing them of their disease. So one of the things that we teach every one of our students is how to use CRISPR for gene editing. We just talked about knock-ins, knockouts, and on this diagram, if you look at the left, basically what you have is a piece of guide RNA. Sort of think about it on whether you're using word processing program or you're searching through your email, you put in a search term. That's that guide RNA. Basically, it searches all 3 billion nucleotides of your DNA until it finds a match. Once it finds that match, it binds. Once it binds to that piece of DNA, then you have the Cas9 cuts that section out. So now you have this gaping hole in your DNA. Now, if you don't fix or you don't have any material to patch that hole, you basically just splice the two ends that you cut together, and now you've cut out whatever genetic material that was there. That's a knockout, so you've just eliminated a gene. Now, if you cut your hole and I put a piece of donor DNA, and now this DNA has the correct gene, now it inserts that correct gene into that hole that you cut, and you now have new DNA inserted into your chromosomes. So you've now corrected a genetic defect. So that's the whole concept behind genetic editing and gene therapy. One of the labs that we do is we take CRISPR, we take E. coli, which is a common garden variety bacteria, and if you put an antibiotic onto the agar plate, the bacteria will not grow. So what we did was we edited so that we could make these bacteria resistant to an antibiotic. Once we made them resistant by knocking out the gene that allows them to be killed by the antibiotic, you can see each one of these spots on this agar plate is a colony of bacteria that are resistant to that specific antibiotic. So we learn how that's done, and that can be applied to anything, not only bacteria, to plants, or to animals. Now, DNA fingerprinting, we hear that's probably one of the things we hear most about, and because different individuals carry different alleles within their DNA, and most alleles are useful for DNA fingerprinting differ in the number of repetitive sequences. So everybody in the world is unique. And if you use those different unique repetitive markers for different individuals, they can identify specific individuals. The very first time this was ever used was in 1983, and it was used in the United Kingdom in a murder rape case, and it was used. This was the first time that genetic fingerprinting was used in a criminal case. In the US, it was 1987 was the first time it was used, again, in the Forest Hills rapist case. Now, a lot of you have heard, and if you like crimies, you will hear this all the time, all the crime shows, I just sent this blood sample to CODIS looking for a match. So the CODIS stands for the Combined DNA Index System, and it's based on, out of all of the thousands of these repetitive elements that exist in our DNA, there are 13 that were found that if you match those 13, the chances of it being two different individuals is 1 in 1 billion. So basically you have a better chance of winning Powerball than you have of finding a second match. So how do we use that? Not only can you use it for determining if you want to send your samples out to 23andMe or if you want to determine paternity. One example, this was used in September 11th. We had over 3,000 people who were burned beyond recognition. And so DNA techniques were used to identify the remains of these victims. Basically using DNA that we derive from bone marrow, you get samples from any family members who'd filed missing person reports using cheek swabs. And then they looked at either short tandem repeats, mitochondrial DNA, or what are called single nucleotide polymorphisms. And using those techniques, they were able to match the family member samples that were provided with the 3,000 victims from the World Trade Center. And it took about 48 hours from beginning to end for them to identify all 3,000 people. Last September, there was a boat fire out in Santa Barbara, basically on Santa Cruz Island, where again, you had some 30 people who were burned beyond recognition. To identify them, they could use what's called rapid DNA technology. So same kind of technique. We make sure this is now becoming the sort of the gold standard for DNA analysis that's used by the FBI and the Homeland Security. This rapid DNA, basically within 90 minutes, you can do an entire sequencing of a particular individual and trying to match it based on these tandem repeat elements. So all of our students learn all of these techniques. This is the Coda system. These are the specific 13 sites that are recognized or by that Coda system. Even though there are more than 100,000, statistically, these 13 are the only ones we need to look at. So we have our students do it. Basically, we have them look at the D1S80. That's one of the 13 alleles. We have all the students take their own blood, look at their own alleles so they can say, okay, that's how I match. We also have them isolate the mitochondrial DNA. Again, it's another marker that's used for identifications. So they actually not only you see it, but you do it, and you know why this works in terms of identifying specific individuals. Now, biotech created therapies. I mean, you cannot turn on the TV without hearing about the coronavirus. And this is all coming from biotech created therapies. Now, some of you may be interested in pursuing either medical school or some other career in healthcare. Well, I show you this picture because this is where my daughter works. She is a physician. She is now a faculty member at Texas Children's Hospital as part of the Baylor College of Medicine. And she spends a couple of days a week there at the place in the middle, MD Anderson, at a place called the Center for Advanced Therapeutics and Genomics. And she uses biotechnology every day. They can't treat patients without using biotechnology. One of the things that she did in her very first publication was looking at developing CAR T cells. CAR T cells, that's a chimera antigen receptor T cell therapy, sort of the newest therapy for treating patients who have tumors which are resistant to common chemotherapy. Basically, it involves they take a blood sample from a patient, they extract the T cells, and then they add a new gene using a retroviral vector into those patients' T cells. They now express this new antigen or new receptor on its surface, and they are now a warrior T cell. They inject back into the patient, and they destroy the tumor. Basically, what they do, normally T cells have what are called this TCR or T cell receptor complex. The alpha and the beta are what recognize the antigen on the tumor. Well, the patient has a tumor, so clearly that didn't work. So what they came up with the idea was instead was, well, what if I take an antibody? And if I take the piece of the antibody which specifically recognizes an antigen and put it onto the T cell and use all the other components of the T cell receptor, then I can now have changed the specificity of that T cell so that it recognizes the tumor. So that's what they did. This is an example of what the retroviral vector looks like. These are transferred into the T cells and with this vector, and it expresses that chimera receptor, and it's expressed on the surface to interact with the tumor. Does it work? Well, here's an example. The diagram says before treatment, this is a patient. All the little black dots are the presence of little micrometastases in tumor. If you look down at the bottom, you see those little yellow areas. Those are tumor masses within the abdomen of this patient. And after treatment, basically 15 months post-treatment, what we see are all the little black dots are gone. You look at the bottom, the yellow dots are gone. This patient is now in complete remission. And this is 15 months after receiving these CAR T cells. And these were patients who failed chemotherapy. So this was their only hopes, which allowed them to use this treatment. So it's a very effective treatment. Molecular diagnostics, that's a really hot field now. We're even seeing that with the coronavirus. But I'm going to tell you about, this is one of the 17 patents which I hold. It's called like a nanosite. It's a nanoparticle tracking analysis. The idea being is if you sit in front of a computer screen or in front of a window with bright sunlight coming in, you see dust particles. You don't really see dust because dust is too small. So our idea was these nanoparticles, they're derived from tumor cells, but they're small. They're about 1,000 times smaller than the diameter of a human hair. So you can't see them without an electron microscope. Since most people don't have an electron microscope, our thought was, well, if we shine a bright light through a solution that contains these microvesicles, then it will reflect the light. We'll be able to see the vesicles. If we see them, then we can count them. If we can count them, then we can tell other characteristics about them. And so this is an example of what we did. This is a sample. We took a blood sample from a woman who had ovarian cancer. We isolated, removed the cells, and we're looking at just the cell-free blood from these patients. And if I get the little, oh, there it is. This is actually a movie. You can see each one of those specks is a vesicle. You can see how there's sort of a rippling effect because they have Brownian movement. We can calculate the size and density of each one of these nanoparticles. So we count them, tell you the size and the content. We can then plot that. And if you look at patients, healthy women who have no sign of any kind of cancer or benign growth, they don't have it. If you look at women who have benign disease, we see a small little peak of material. But women who have ovarian cancer, we see this huge peak of these nanoparticles that are circulating in their blood. So this is a quick, it takes about 10 minutes from beginning to end assay for the detection of ovarian cancer. Now, the last thing that I always love is cloning. I keep wanting to clone dinosaurs. Jurassic Park is one of my favorite movies. This is a picture of Dolly and Dolly's mom. She was created using what's called nuclear transfer. They took an egg from a female sheep in this case, and we took the nucleus from the mom that was going to give birth to or give rise to Dolly. We take out the nucleus and take that nucleus and stick it inside of the egg. So now you have what's sort of shown there on the right, where you have this new nucleus inside the egg. You implant it into the surrogate mom, and then in a few months you have the birth of Dolly. This is called transferring of a nucleus. In this picture here, you see the cute little lamb on the side. That's Dolly when she was born. She was born in 1996. Has her mom beside her. Her mom was 7 at the time that she was born. Dolly died in February 14th of 2007, basically lived about 7 years, and that's half the normal lifespan of the sheep because the nucleus was already 7 years old when you took it out and put it in the egg. So she was already, even though she's a cute little lamb, she was really, her cells at the cellular level, she was 7 years old. So as a result, she died of old age, and her death was due to progressive lung disease and early arthritis resulting from her cells already being aged, which is one of the reasons why in all the sci-fi movies, when you never see these people dying at early age, but this is one of the considerations one has to look at when they're cloning. This was the first cloned pet. This is called carbon copy. This is all of the carbon copies, basically all of this is rainbow and carbon copy. These are 2 clones from the same original mom. Like I said, I keep wanting to clone dinosaurs, but the faculty or the administration is a little bit antsy about cloning something that may eat our students. So instead, last year we cloned an African violet. And so this is sort of an example of what we cloned, and students were able to take them home to their parents at the end of the school year. So it's kind of a new sort of approach. Instead of saying, hey, mom, look what I made for you in school, you can actually say, look what I cloned for you in school. So it gives you a sort of a different, but it's all the techniques are the same, whether you're cloning a plant or an animal, they're all pretty much the same technique. So where I want to sort of end this little topic before I take your questions is, if this is our friend, or not so much friend, COVID-19. So this is what it looks like when you stain it and look at it under the electron microscope. And if you say, well, how do I know if I've been exposed to it? What are the symptoms? Well, if you have severe infection, you may have a fever, a cough, shortness of breath. Now, that's pretty much the same symptoms you have with the flu, or allergies, or any of a number of conditions that are not COVID-19. So how do you know? Well, you're hearing a lot about different types of tests. The most accurate are what are called endpoint PCRs or real-time PCR tests. Here's the examples of 2 of those. The one that's a little bit more cumbersome, it's also been done when they've been tracking what cases may have originated in China and which ones may have come from China via Europe into the United States. These are all done, and these identification of mutations within the COVID-19 are done by this virus DNA sequencing chips. You also will hear a lot about antibody techniques or these antibody tests. Here are 2 that I show you on the right, basically the rapid immunodetection and the COVID-19. This is an IgM, IgG antibody test. Downside of it, so you learn, as students, as our program, we do a lot of endpoint PCR. We also do a lot of real-time PCR. We don't do it on viruses, but it gives you the knowledge of the techniques which are currently being used in the industry. Sequencing, we do sequencing. We're not going to sequence any viruses, even though we do have a biosafety level 2 facility. It's not suitable for handling viruses, but we can handle human-derived materials. And these rapid immunodetections using antibodies, these are new tests. We go through this even for next year for the lab. One of the labs we have is we're going to take you through one of these antibody tests, and the lab is going to be set up to sort of a spin off on The Walking Dead. We're going to identify patient zero using a antibody test for monitoring or screening through patient zero from the zombie apocalypse. So that just sort of gives you an idea of the kinds of things, directions we're going, and what biotechnology can do for you. And at that point, we can open up any questions that you might have about our program, about any of the specific things that we may be learning, any questions, concerns you may have about the program in these very odd times. Any of those can be fair game. So with that, we can open up the questions.
All right. Thank you very, very much, Doug. You do such a wonderful job of taking what I think is some pretty complex information and explaining it to us in layman's terms. So I hope that our audience feels the same way. We do have some questions. We have a couple sitting here waiting in the queue. We also had some questions that were sent in prior to today's event. So I'm going to be kind of going back and forth here and hitting you up with a bunch of stuff. So we have a question here from our audience from Samuel Walker. He wants to know if you can enroll in multiple biotechnology courses, such as general biotech and nanotechnology. So he wants to know if you really wanted to take both concentrations, is that a possibility or does that make sense?
It makes sense and it depends on your interest, but the short answer is yes, you can. And we have a number of people who will be major in two different focus groups. More than two is a little bit difficult and still get it done in 4 years, but we have I'd probably say at least a third of our students end up majoring in two different focus groups. You know, there's the general techniques that you want to learn, and then sometimes you think, oh, you know, I really am interested in food science, or I'm really interested, like in this case, in nanobiotechnology, because a lot of the new tests, you know, some of the ways they're doing some of these vaccines, the University of Pennsylvania, their new vaccine for COVID-19 is they're putting DNA on gold nanoparticles, and then they're using gold nanoparticles to bombard the cells of the, you know, as part of a vaccine. The DNA is taken up by the cells, it's expressed, they create the antigen, express it on the surface, and it generates an immune response. So their vaccine is based off of sort of a combination. The vaccine, you isolate the DNA, which would be a general technique, and then coupling it to a nanoparticle, which would be under the nanoparticles, nanofabrication, nanobiotech technology. So yeah, some of those things like that fit really well together. So yeah, that's an easily doable double focus.
All right, wonderful. Sam actually has a follow-up question, and he asks, you know, if he wanted to get into doing vaccines for a living, what would be your recommendation if you could give him one of which concentration might be the best for him?
So my recommendation for that would be the general biotechnology. We actually start with, you know, in the first part, one of the first classes that you'll get will be what's called biotechnology techniques, where we take you through every step that you will have to use to create or isolate the materials to make the vaccine with. Then how do you genetically modify, or depending upon how you want to do you want to make it your vaccine in a plant? Things like ZMapp, the vaccine for Ebola virus, was made in tobacco plants. So if I want to transfect it into a tobacco plant, how do I do that? So that would be, again, a technique that's learned in general biotechnology. And then if you say, well, actually, I want to express just a particle of that, and I want to make it in a bacteria. So these are what are called single chain variable fragments. They're commonly used as sort of an antibody or a version of antibody technology. Again, that would be it's a form of a vaccine, but again, that's part of the general biotech major.
Thank you very much, Doug. We have another question here from Ashley, and this actually was asked by quite a few people prior to the event and has to do with medical school. So Ashley's specific question is, would you recommend a student to major in biotechnology if they want to become a future physician? So if you could speak to that a little bit, Doug, as well as maybe share with our audience some of the schools that our graduates have actually been accepted to and attended as medical students or pharmacology or whatever it might be?
Okay, so start with the short list. So if you were interested in medical school, should you do biotech? Like I said, my daughter is a physician, and I hear every time I talk to her, she wishes she had majored in biotech and not biology, because a lot of the things that she does, she's having to relearn them, and she's constantly like, "Could you send me your lecture on this so I can go through it? Because I didn't learn this." And you're like, "How do you do CRISPR? How do you do, this ELISA technique? How do you develop a, isolate a piece of DNA in order to couple it to a nanoparticle?" Those are things they just don't teach in biology. So when you think about, if you were, say, a biology major, and you say, okay, I'm going to learn that cancer is caused by a mutation and it allows uncontrolled growth. In biotech, you would say, okay, you learn exactly the same thing, but then you take it one step further and say, okay, how can I develop a diagnostic test that detects the mutation? Okay, then how can I develop a drug that will target the result of that mutation? And then ultimately, how can I develop a gene editing or gene therapy to repair the mutation that caused the cancer? So you learn the same material plus the application of how do you fix it, which is really what physicians are all about. Is not only knowing that you have it, but fixing it. And when you look at some of our students where they've gone, we have like a number of our biotech students have ended up, we have one that went to Princeton, is working on a PhD. We have one from last year. She is down at Duke working on her PhD in biotechnology. We have one of our students this year is going to the George Mason University, where they're going to be doing a master's in biotechnology. We have one that's going to Penn State to work on their master's degree in biotechnology. We have one that's at the University of Maryland, and because they're interested in medicinal marijuana, so they're interested in the development or genetic alterations of cannabis plants. And they actually have a program, a master's degree in cannabis science. So they're going down there to pursue a master's degree. So we have a lot of students that go out that pursue PhDs, and we have some that go out and pursue medical, or they're going into medical school, but been based off the technologies because of the specific applications. A lot of people who are interested in things like Alzheimer's disease, Parkinson's disease, any kind of neurologic defect, because we do have kind of a unique feature. I have sort of an interest, or I have an interest in neurology. The last patent that I filed for before we sold our company was a test for looking at concussions and development of chronic or CTE, and with football players. And so our students get sort of a unique application. So how can biotech be applied to the brain, especially in terms you can't biopsy it? So how do you assess it? How do you create, computer brain interfaces? So a lot of those things, again, those all apply to the medical field.
Wonderful. We have another question just very quickly to bounce off of that. We have someone in the audience who wants to know if taking a biotech major would also be applicable to veterinary school. Would you say that it's really the same thing, or any reason why it wouldn't?
No, I'd say it would be exactly the same thing. Plus, just sort of as an interesting tidbit, some of the things, because I'd come from the state of Kentucky originally, and we should be running this week. We should be having the Kentucky Derby, but this is an odd year, so we're not. One of the applications that people have made of biotech is that you can take, when they go to the yearling sales in Kingland for horses, is like, how do you know before I spend millions of dollars on this yearling who can hardly walk, much less run? How do I know if they have the potential to be a Triple Crown winner? Well, there are a number of genetic markers that you can look at that will define characteristics such as those that are fast versus how that have longer endurance. And so that's really turned into a science of using biotechnology to look at specific characteristics of animals, as well as determining what their potential is in terms of marketable animals. So it has, and all the drugs that we develop, all of them again come through biotech pathways. So it's kind of the same thing, and it's still regulated by the FDA, which is sort of why it uses the same pathways.
Okay, awesome. We have so many more questions coming in. We're getting down to the wire here. I'm going to try to get as many in as I can. A couple different questions here just about careers, but I'm going to focus on this one. We have another anonymous person in the audience says, is a master's degree required to get a good job in biotech after college, or is the bachelor's degree enough to get a good job in Pennsylvania?
It's a good enough, basically a bachelor's is good enough to get you a good job in Pennsylvania or pretty much any other state. The beauty of it is, and the way we've designed the program here, there is what's called the National Center for Biotechnology Education. They have a checklist that says these are the gold standard skills that you need to have to be employed in the biotech industry. And so we have developed our program looking at their checklist to make sure that when you graduate, you've checked off every technology on the list so that you are employable. Now, the beauty of it is that, and we have a lot of our students that do this, is they graduate and then they'll go out and get a job. Say maybe they'll go out to Lancaster Labs and they get a job developing cell-based assays. So they're doing that 9 to 5. Then in the evening, they may say, you know, I've been doing this for a year. I've been paying off whatever loans, or I've been making a living. It's kind of nice not to be, you know, living in mom and dad's basement. But, you know, now I want, instead of working in the lab, I want to be the manager of the lab. So we have these people now say, you know, at night I can take online classes and get a master's degree in biotechnology, more in supervisorial skills, how to troubleshoot, how to fill out all of the regulatory paperwork so that you can have a master's in biotechnology. There are multiple schools, including Harrisburg University, that offers you an online master's degree so you can work during the day and still get further education at night. But a bachelor's degree is really all you need to get your foot in the door at a high-paying job.
Yeah, and who knows, sometimes your employer will help pay for that master's degree, which would be nice. So
that happens. That is very common, that they will pay for, the employer will pay for that.
Right, wonderful. We had a couple of different questions that were presented before today's event, and I'm going to paraphrase them. You know, I know biotechnology is sort of like the application of biology, I guess you could say. There seems to be a lot of overlap in some of our other majors. For example, forensics. There seems to be some overlap when we talk about, you know, the DNAs and the sampling, all that kind of stuff, crime scene things, as well as maybe in advanced manufacturing when it comes to maybe actually the robotics part and putting together these applications. Do you agree with that? Is there overlap between the majors, and do you work with other departments on projects together?
The answer is yes. I mean, basically the sort of downside, currently the forensics program at HU is purely from a crime scene perspective. There isn't any, all of the DNA technology is exclusively, and biotech even offers multiple classes in the DNA and molecular forensics. Basically, how do you sample a DNA, or how do you collect a DNA sample? How do you extract the DNA? How do you extract mitochondrial DNA? How do you analyze for single nucleotide polymorphisms? There's no courses in forensics that handle any kinds of technology like that. So those are really exclusively in the area of biotech. So if you say, I want to get a job doing DNA forensics, then biotech's the only program that offers those classes. If you want to know how to put tape down, or how to manage a lab, how to fill out the paperwork, how, you know, sort of the nuts and bolts of a crime scene, then forensics is the area to go. But if you actually want to, how do I do the laboratory work, the nuts and bolts analysis of DNA and RNA, then that's really a biotech company. But then with the advanced manufacturing, yeah, it's kind of like there's the device and there's the interface. And so as a result, we're sort of that was something we actually combined together, because it's really the advanced manufacturing. You make something, and then over here is how do you use it? So it's sort of those two applications come together between advanced manufacturing and biotech in terms of medical devices.
All right. Well, Doug, thank you very, very much. I wasn't able to get to every single question, but I really honestly think that a lot of the questions that were posed were answered in other ways. So I hope everyone out there really, really enjoyed this today. Like I said, Doug, you do a great job of explaining everything to everyone, techie or not techie. Very interesting stuff. This was recorded, and we will be sending out a link to the recording to every single person who registered for this event. You are more than welcome to share it with anybody and everybody. And I did put Professor Taylor's name and email address out at the beginning of the presentation. If you missed it, I'm going to quickly put it there again, but his name is Douglas Taylor, and his email address is dtaylor, Taylor, at harrisburgu.edu. So with that being said, we're going to bid you all adieu and wish you a very wonderful day, and we hope you join us again. Our next webinar is this Thursday. It's all about student support. It's really geared for students who are about to graduate high school or thinking of transferring, and their parents. So we look forward to seeing some of you again on Thursday, and keep an eye on your email and cell phones for more invitations. Thank you and have a great afternoon.